The human body is about 15% by weight protein. In this video, we will explore the structure of proteins. Proteins are large molecules known as macromolecules. They are made of individual subunits called amino acids. Each of these blue circles is supposed to represent an amino acid, which are bonded together to form what we call a polypeptide chain. If that chain is long enough, we refer to it as a protein. Some proteins have hundreds of amino acids along a chain. We begin our study of protein structure by studying the structure of the building blocks of proteins, the amino acids. At the core of an amino acid is a carbon atom. This means we're dealing with an organic compound. Recall that carbon can form 4 covalent bonds. So attached to that central carbon are 4 groups. One of the groups is going to be an amino group, NH2. The dash implies a covalent bond. Amino implies it contains nitrogen. A second group attached to the central carbon is a carboxylic acid group, COOH. And that's where we get the acid in amino acid. An amino acid has at least one amino group and one acid group as part of its structure. The third group is just a hydrogen atom. All amino acids that are used to make proteins in our bodies have these 3 groups in common. What makes one amino acid different from another is the 4th group. The 4th group is the variable group, sometimes called the side chain. Usually it's an organic group, although there is an exception. In this generic amino acid, we have our central carbon. To the central carbon will bond 4 groups. One of the groups is a mere hydrogen atom. Next, we'll add the amino group and bond it to the central carbon. Now we'll bring in the carboxylic acid group and bond it to the central carbon. The 4th group, however, varies from one amino acid to another, so I've symbolized this group by the letter R. The R group is also called the side chain. Amino acids differ in this 4th group, which accounts for their different behaviors. Now let's look at some examples of amino acids. The simplest of all the amino acids is glycine, whose side chain is merely a hydrogen atom. All the amino acids have a 3-letter designation, usually the first 3 letters of their name. The 3-letter abbreviation for glycine is GLY. The backbone of an amino acid is what they all have in common, and is shown here in yellow. The unique side chain of glycine is simply a hydrogen. So this is the one amino acid that does not have an organic group as its 4th group. Alanine is the next simplest amino acid. Its side chain is a methyl group, CH sub 3. Other amino acids have more complex side chains. Often you see amino acids grouped based upon similarities and differences in their side chains. Some amino acids have side chains that are not water-soluble. These are called nonpolar side chains or hydrophobic side chains. They include the amino acids phenylalanine and leucine. Notice the amino acid phenylalanine has a hydrocarbon side chain which includes a ring structure. Sometimes this is called the benzene ring structure. It's not shown, but at each corner of the ring is a carbon. There's actually 6 carbons in this ring, and attached to those carbons are hydrogens. They're not shown either. The side chain of phenylalanine is not electrically sticky, which means that water has no way of grabbing hold of it, and so it's not very water soluble. It will tend to avoid water or be pushed away from a watery matrix. On the right, you've got another amino acid with a nonpolar side chain. This is leucine, abbreviated LEU. Notice that it has a hydrocarbon side chain which ends in 2 methyl groups. A methyl group is a carbon bonded to 3 hydrogens. This is sometimes called a branched chain amino acid because it forms 2 branches composed of methyl groups. Some side chains are water-loving or hydrophilic. Phil means love. Philharmonic means love of harmony. Philadelphia means brotherly love. Philanthropy means the love of humanity. The side chains here are more water soluble. They are electrically sticky, and so water has an affinity for them and can dissolve them. On the left is serine, abbreviated SER. Notice that serine has a hydroxyl group at the end of its side chain. On the right is tyrosine. Tyrosine is similar to phenylalanine because its side chain includes a benzene ring. But what makes tyrosine more water-soluble than phenylalanine is the hydroxyl group that hangs off the ring. The addition of that hydroxyl group makes tyrosine more water-soluble than phenylalanine. 2 amino acids have sulfur in their side chains, cysteine and methionine. The sulfur atom in a cysteine side chain can form a covalent bond with the sulfur in a second cysteine in a protein. These disulfide bonds can help fix a protein in a specific 3-dimensional structure. Here's a list of the 20 standard amino acids that are used as building blocks of the proteins of our body. 9 of them are shown in red because they are essential in the diet. Those shown in yellow can be manufactured inside the body. For example, phenylalanine must be obtained in our food. But if we have enough phenylalanine in our diet, we can use some of it to make tyrosine by adding a hydroxyl group to its side chain. There are many more than 20 amino acids found in nature. There are more than 20 amino acids in our body. But our genetic code specifies these 20 amino acids. And so we consider this group to be the standard 20 amino acids needed to construct the various proteins of our body. These 20 amino acids are the alphabet of the proteins of our body. By stringing these amino acids together in various combinations and in various lengths, we can manufacture the various proteins that our body needs to do its work. Some of these amino acids are modified after they're incorporated into proteins. Nevertheless, these are the foundational 20 coded for by the genes inside our cells. Of course, an amino acid itself is not a protein, any more than an individual letter of the alphabet is a paragraph. So if we're going to make a protein, we need to covalently bond amino acids together to form a chain. And that's what we'll do now. Notice at the top, on the left, we have the amino acid glycine, the simplest of all amino acids, because it has only a hydrogen as its side chain. Notice on the right we've got the next simplest amino acid, alanine, which has a methyl group as its side chain. What we're going to do now is bond these two amino acids together to form a dipeptide, the first step in producing a protein. If we're going to form a covalent bond between these two amino acids, we're going to have to break bonds in each amino acid in order to free up bonding potential. Recall that carbon can form only 4 covalent bonds, and glycine already has 4 bonds on the carbon atom in its carboxylic acid group. 1, 2, 3, 4. Nitrogen can form 3 covalent bonds. The nitrogen in alanine already has 3 covalent bonds. 1, 2, 3. So these amino acids have no more bonding potential. For them to bond to each other, we must break some bonds first. We're going to break a bond in glycine that holds this carbon to this hydroxyl group. And we're going to break the bond on alanine that holds this nitrogen to this hydrogen. And by breaking those bonds, we open up a bond potential between this nitrogen and this carbon. And so the carbon and the nitrogen can form a covalent bond with each other, thereby merging to form a dipeptide. The bonds that holds these amino acids together is called a peptide bond. Notice that when we remove the hydroxyl group from glycine and the hydrogen from alanine, we created water, H2O. When we form a peptide bond between 2 amino acids, one of the byproducts is water. We produce water every time we add a new amino acid to the growing polypeptide chain. The formation of peptide bonds is an energy-requiring process. It requires input of energy in the form of ATP. So one of the reasons we eat is to get the energy needed to build the proteins inside our body. Here's an animation of what happens in a peptide bond. They're highlighting the acid and amino groups of these 2 amino acids. Notice that to bond the amino acids, we must first break bonds, making water. Then we bond the nitrogen of one amino acid to the carbon in the carboxylic acid group of the second amino acid. The bond that links the 2 amino acids together is a peptide bond. The combined structure is called a dipeptide. If we add a 3rd amino acid, we make a tripeptide. If we add a 4th, we make a tetrapeptide. And a 5th, we make a pentapeptide. After a while, we run out of prefixes, and so we say we have a polypeptide chain. Or if the chain contains more than 50 amino acids, we call it a protein. And each time we form a new bond between amino acids, we make more water. Let's make a polypeptide chain. Notice each time we form a peptide bond between 2 amino acids, water is a byproduct. So in linking these 5 amino acids together, we produced 4 water molecules. Here we're making a tetrapeptide. It contains 1, 2, 3, 4 amino acids. In forming these 3 covalent bonds that hold these 4 amino acids together, we produce 3 water molecules. An adult will produce about a cup of water a day inside the body as a result of chemical reactions like protein synthesis. Here's an example of a simple protein. Each one of these blue circles represents an individual amino acid, and each one of these circles are labeled as a particular amino acid. The primary structure of a protein is the linear sequence of amino acids, beginning with the free amino end and going to the free carboxylic acid end. There it is. If you were asked to describe the primary structure of this protein, you would start here and begin listing the amino acids in the order of their appearance. We would say lysine, valine, phenylalanine, glycine, arginine, cysteine, glutamate, leucine, alanine, alanine, methionine, and so on, all the way along the chain till we reach the free carboxylic acid end. This particular protein ends with the amino acid leucine. So a simple protein is a long chain of amino acids covalently bonded together. Here's a very brief polypeptide chain. If I were to ask you to spell out the primary structure of this protein, you would begin at the free amino end and say methionine, tryptophan, aspartic acid, isoleucine, glutamate, valine, serine, phenylalanine, asparagine, arginine, glutamine, and lysine at the free carboxylic acid end. This is what we call the primary structure of a protein. Proteins are more than a linear chain of amino acids. They have higher order structures. Here's an example of a protein with a couple of different secondary structures. Twice you can see that the amino acids that are near each other form helical structures, here and here. The helical structure is known as a secondary structure. In some proteins, the polypeptide chain may fold back on itself to form sheets called pleated sheets, like you see here. This would also be considered a secondary structure. It's as if you took a chain of beads and folded it back on itself to form a sheet. The secondary structures of proteins are due to the interaction of nearby amino acids. Many proteins have a third level of structure known as a tertiary structure. This is due to the interaction of the side chains of amino acids located distantly along the polypeptide chain. And this produces large folds in the protein that can create a globular structure or spherical protein. You can see here one of the major glues holding this protein together in its three-dimensional structure are disulfide bonds. These are between the sulfur atoms that are found in the side chain of the amino acid cysteine. There may be other interactions, such as ionic bonding and hydrogen bonding between the side chains of amino acids that contribute to the formation and the stabilization of a protein in this three-dimensional configuration. But the disulfide bonds are the strongest. Typically, the proteins that circulate in our blood are in a globular state. When we need to clot the blood, we convert one of these globular proteins, fibrinogen, into a fibrous protein called fibrin, which forms a net at a wound to stop the loss of blood. Let's examine the different layers of structure in this protein. All proteins will have a primary structure, meaning a linear sequence of amino acids. Notice that this protein also has a secondary structure. See the helical structures here, and here, and here, and here. This protein also has a pleated sheet structure, where the protein has folded back onto itself, forming multiple layers. We also have large folds in this protein, like you see here, that creates what we call a tertiary structure, or globular structure. These different folds are held together by ionic bonds, the attraction of opposite charge, hydrogen bonds, as well as disulfide bonds, bonds between the sulfurs in the side chains of the cysteine amino acids. Some proteins have a fourth level of structure. Probably the most famous example of quaternary structure is that of hemoglobin. Hemoglobin contains more than one globular protein that have coalesced to form this higher order structure. So a protein with a quaternary structure has two or more protein chains composing it. Hemoglobin is composed of 4 globular proteins, 2 shown in yellow and 2 shown in blue. Hemoglobin is found inside the red blood cell, where it's needed to bind and transport molecular oxygen. This fourth level of structure, or quaternary structure, is necessary for the functioning of hemoglobin. The most abundant protein in the human body, collagen, is composed of 3 polypeptide chains wrapped around each other, forming a tough, fibrous protein. Nearly a third of all the protein in our body is collagen, which is found in bones, tendons, ligaments, and the deep layer of the skin. One of the amino acids in collagen, proline, is modified after protein synthesis by the addition of a hydroxyl group, forming hydroxyproline. Vitamin C is needed to hydroxylate proline. A vitamin C deficiency will impair the production of collagen and lead to a disease called scurvy, where one literally comes unglued. Some proteins may have a 5th level of structure, but this 5th level is not actually protein. The 5th level would be non-protein cofactors, such as vitamins acting as coenzymes, or ionized elements, popularly called minerals, like magnesium or manganese or selenium, that bind to the protein and activate it. Once again, you're looking at a model of hemoglobin. If you look closely, you can see 4 organic groups circled in red called heme groups, bonded to the 4 proteins of hemoglobin. Without these heme groups, hemoglobin would be of no use in the transport of oxygen. Here is a summary of the 4 levels of protein structure. The primary structure is the linear sequence of amino acids. The secondary structure is the interaction of nearby amino acids forming helices as well as pleated sheets. The tertiary structure is where distant amino acids interact along the protein chain to cause major folds in the protein, producing a globular protein. The 4th level of structure, the quaternary structure, involves 2 or more proteins that coalesce together to form a higher order structure. Many proteins also have a 5th level of structure, which includes non-protein cofactors that bind to and help activate the protein. Let's see how many levels of structure we can find in this protein. All proteins have a primary structure, a linear sequence of amino acids. And if we were to elucidate the primary structure of this protein, we would start at the free amino end and list the sequence of amino acids along the chain until we reach the free carboxylic acid end. Notice how the amino acids are twisted to form a helix. This is an example of a secondary structure. Does it have a tertiary structure? You can see where major folds occur. So what we have here is a globular protein. Does this protein reveal that it has a fourth level of structure, or quaternary structure? No, there's only one polypeptide chain here, so it has no fourth level of structure. How about a fifth level of structure? Notice this group shown in red. This is a heme group. What we have here is a model of myoglobin, which is a simpler cousin to hemoglobin. The function of a protein arises from its structure. Think of a paperclip. A paperclip holds papers together. But if you change the structure of a paperclip, if you unfold it and make it a straight piece of metal, it no longer functions as a paperclip. You may be able to use it as a fingernail cleaner, or as a weapon on an airplane, or to pick a lock. But you're not likely going to be able to use it anymore as a paperclip. Unfolding a paperclip doesn't change its composition, it changes its structure. But by changing the structure of the paperclip, you can change its function. Drop a frog in a blender and you no longer have a living frog. All the ingredients are still there, but instead of a living frog, you now have frog soup. Something similar is true of proteins. Protein function depends upon protein structure. Enzymes are proteins that act as agents of chemical change in the body. To perform their chemical feats, an enzyme must bind to its substrate. To bind its substrate, an enzyme must present a very specific surface to its environment. Extreme environments may cause enzymes to get bent out of shape, and the result is they can no longer bind their substrate and create their chemical product. If you alter the structure of a protein, you can alter its function. And two ways of altering the structure of a protein would be to heat it or to expose it to strong acids or bases. Let's crack open an egg and drop it onto a skillet. Egg white is composed of globular proteins. Fresh out of its shell, the egg white is liquid and transparent. But when it hits the hot skillet, the heat causes the globular proteins to unravel. And when that happens, the egg white becomes an opaque solid. And all the king's horses and all the king's men can't put it back together again. This is an irreversible reaction, so you might as well eat it. The same thing can happen in our bodies. That's one of the reasons why we regulate body temperature. If our body temperature rises too high, we can fry our proteins. The fancy name for unraveling our proteins is denature. And when our proteins denature, they lose their function. We can tolerate a temperature a few degrees above normal, but when your oral temperature goes up to, say, 107 degrees Fahrenheit or higher, then you're getting to a point where it's going to significantly affect the structure of your proteins. They're going to come unglued due to the increased kinetic energy of their environment. And they may come unglued to a sufficient extent that they won't renature. The result is you could end up dead. On the left here, you see a globular protein. It's held into its three-dimensional conformation by a variety of forces that may include hydrogen bonds, ionic bonds, and disulfide bonds. But if we increase the kinetic energy of the environment of this protein, that protein is going to be jostled around and bounced into other molecules and potentially ripped apart. Not only will heat interfere with the forces that hold proteins together in their three-dimensional shape, but so will a change in the acidity of a protein's environment. If you take egg white and drop it into an acid like vinegar, you'll notice changes in the egg white similar to your dropping it onto a hot skillet. That's why the body also regulates the pH of the body fluids, so that the acidity remains stable. If we allow the pH to go too low or too high, it can interfere with the binding of the side chains of our proteins, causing them to denature, which in turn will impair or destroy the function of our proteins. Protein function arises out of its structure. The structure of a protein ultimately depends upon its linear sequence of amino acids, which in turn determines how the protein folds up, which then determines how the protein functions. Hemoglobin has 4 polypeptide chains in globular form linked to each other. On this diagram, 2 of the chains, known as alpha chains, are shown in yellow, and the other 2 chains, called beta chains, are shown in blue. The sequence of amino acids in the 2 alpha chains is the same. So the 2 alpha chains can be coded by the same genes. If you know the sequence of amino acids in one alpha chain, you know the sequence of amino acids in the second alpha chain. If you know the sequence of amino acids in one beta chain, you also know the sequence of amino acids in the other beta chain. So it really takes only 2 genes to code for the 4 proteins that are found in hemoglobin. To function, hemoglobin has to have this particular conformation. One reason why hemoglobin is found inside the red blood cell is that the red blood cell provides an environment that is hospitable for hemoglobin to maintain its shape and therefore maintain its function. If the red blood cell were to rupture and hemoglobin were to spill out into the plasma of the blood, it would quickly lose its function. That's why we can't just inject hemoglobin into people who've lost a lot of blood, because the hemoglobin is unstable unless it's inside the red blood cell. The red blood cell creates a protective environment for the hemoglobin molecule to do its job. And that job, of course, is to bind molecular oxygen. Hemoglobin binds oxygen at 4 sites: the heme sites, the non-protein sites that are present in the hemoglobin molecule. When we inhale, we deliver oxygen to the air sacs in the lungs. The oxygen diffuses into the blood and into the red blood cell, where it binds to hemoglobin. Oxygen is not very soluble in water, and hemoglobin increases the oxygen-carrying capacity of the blood by as much as 70 times. The oxygen is distributed to the skeletal muscles and other organs of the body, where the hemoglobin releases the oxygen, making it available to the cells to manufacture ATP, the universal energy currency. Even a minor deviation in the sequence of amino acids of a protein like hemoglobin can have disastrous consequences. Each beta chain of hemoglobin contains 146 amino acids. The first 6 amino acids of a normal beta chain are valine, histidine, leucine, threonine, proline, and glutamate. There is a genetic disease that results in the substitution of one amino acid for another in the beta chain. The disease is called sickle cell anemia and involves the substitution of the amino acid valine for the amino acid glutamate in the 6th position of the beta chain. The remaining 145 amino acids in the beta chain are the same. And yet, as a result of that one substitution, we get a genetic disease that is life-shortening. This small change in the sequence of amino acids causes the hemoglobin molecules to clump together when they unload oxygen. This distorts the red blood cell into a sickle shape. These sickle-shaped cells can get stuck in the tiny capillaries, causing anemia, a lack of oxygen delivery to the cells resulting from defects within the red blood cells.